Alpha Lifetech Peptide Library Platform. Peptide Library Introduction. A collection of numerous peptide sequences, typically varying in length from 7 to 20 amino acids, generated through combinatorial synthesis. These libraries can contain billions of unique peptides. Cyclic Peptide Libraries. Cyclic peptide libraries are collections of peptides in which each peptide has a cyclic structure, rather than the linear structure typically found in conventional peptides. The cyclic nature of these peptides confers several advantages, in terms of stability, binding affinity, and resistance to enzymatic degradation, making them valuable tools in drug discovery. Cyclic peptides are peptides whose amino acid sequences are covalently bonded in a loop, forming a cyclic structure. Disulfide bond cyclization: cysteine residues within the peptide form disulfide bridges, creating a loop. The cyclization can occur through various chemical bonds, such as peptide bond cyclization. The N-terminal amino group and the C-terminal carboxyl group of the peptide form a peptide bond, closing the chain into a ring. Side chain to side chain or side chain to backbone cyclization: non-standard amino acids or modifications enable cyclization through covalent bonds between side chains and the peptide backbone. Peptide library display platforms. Phage display, the most common platform, where peptides are fused to the coat proteins of bacteriophages, such as M13 or T7. The phage displays the peptide on its surface, while the DNA encoding the peptide is contained within the phage, linking genotype and phenotype. Yeast display: peptides are fused to surface proteins of yeast cells, enabling flow cytometry-based selection. Ribosome display: in vitro translation system where peptides are linked to their encoding mRNA via the ribosome, facilitating direct selection of peptides without the need for a living host. mRNA display: peptides are covalently linked to their encoding mRNA, enabling in vitro selection processes similar to ribosome display. Cell surface display: peptides are displayed on the surface of bacteria or mammalian cells, allowing for screening in more physiologically relevant environments. Peptide library screening introduction. Peptide library screening used in drug discovery, molecular biology, and protein engineering to identify peptides with specific binding affinities, enzymatic activities, or other desirable properties. This process involves creating a vast collection of diverse peptides and testing them to find candidates that interact with a target molecule, such as a protein, nucleic acid, or small molecule. Key steps in peptide library screening. Library construction. A peptide library is generated, typically through combinatorial synthesis, where a large number of peptides with varying sequences are created. These libraries can be displayed on various platforms such as phage display, yeast display, ribosomes, ribosome display, or synthesized directly for screening in solution or on solid supports. Target preparation. The target molecule, which could be a protein, enzyme, receptor, or other biomolecule, is prepared and immobilized on a solid surface or kept in solution depending on the screening method used. Binding assay. The peptide library is exposed to the target molecule. Peptides that have high affinity for the target will bind to it, while non-binding or weakly binding peptides are washed away. Selection of binding peptides. After incubation, the bound peptides are isolated from the unbound ones. This is typically done by washing the target-bound complexes, ensuring that only strong binders remain attached. Elution of bound peptides. The bound peptides are then eluted from the target. This can be achieved through changes in pH, ionic strength, or by competitive binding using a known ligand or antibody. Amplification, if applicable. In cases where the peptides are displayed on a platform like phages, the eluted peptides can be amplified by infecting bacteria, for phages, or by PCR amplification of the encoding DNA/RNA sequences. Each round improves the quality of the binding peptides by removing non-binders and weak binders. Screening and sequencing. After several rounds of selection, the enriched peptide pool is sequenced to identify the peptide sequences that have high affinity for the target. Modern sequencing techniques, such as next-generation sequencing, NGS, allow for rapid and comprehensive analysis of the selected peptides. Validation and characterization. The identified peptides are synthesized and subjected to further testing to confirm their binding affinity, specificity, and functional activity. This might include binding assays like surface plasmon resonance, SPR, enzyme-linked immunosorbent assays, ELISA, or functional assays in cell-based systems. Applications of peptide library. Drug discovery. Identifying peptide inhibitors or agonists for therapeutic targets, leading to the development of peptide-based drugs. Epitope mapping. Determining the specific regions of antigens that are recognized by antibodies, aiding in vaccine development and immunodiagnostics. Protein-protein interaction studies. Discovering peptides that can disrupt or mimic protein-protein interactions, providing insights into cellular pathways and potential drug targets. Key features of a custom peptide library. Design flexibility. Amino acid composition. You can choose the specific amino acids included in each peptide, which is useful for focusing on certain properties like hydrophobicity, charge, or structural motifs. Peptide length. The length of the peptides can be customized, ranging from short sequences, example, 5 to 10 amino acids, to longer peptides, example, 20 to 30 amino acids or more. Sequence variations. Custom libraries allow for the introduction of specific mutations or variations in the peptide sequences, which is critical for exploring structure-function relationships. Diverse library formats. Linear peptides, straightforward sequences of amino acids, useful for basic binding studies and epitope mapping. Cyclic peptides, peptides with covalent bonds forming a cyclic structure, often used to improve stability, binding affinity, or resistance to proteolysis. Modified peptides, incorporation of non-standard amino acids, post-translational modifications, like phosphorylation or glycosylation, or labels to suit specific experimental needs. Synthesis methods. Solid-phase peptide synthesis, SPPS, a common method for synthesizing custom peptides where peptides are built step by step on a solid resin. Parallel synthesis. Multiple peptides are synthesized simultaneously, allowing for the rapid creation of a diverse library. Alpha Lifetech can provide Phage display peptide library display services and peptide libraries inventory. Peptide library includes 12 peptide libraries: pentapeptide library, dodecapeptide library, heptapeptide library, cyclic heptapeptide library, linear 9 peptide. Peptide library includes 12 peptide libraries: pentapeptide library, dodecapeptide library, heptapeptide library, cyclic heptapeptide library, linear 9 peptide.